our research program focuses on a cellular process that is critical for the spread of cancer, or metastasis. Cancer starts with mutations in our DNA, which leads to uncontrolled proliferations of cells. These tumor cells can spread to other parts of the body where they can form secondary tumors. The spread of cancer is also called metastasis. About 90% of deaths from cancer are caused by metastasis. In breast cancer, for example, the prognosis of patients with metastasis is much worse than those without metastasis. In many types of cancer, metastasis starts with a dramatic change of epithelial cancer cells. These cells become invasive in this process. They can get into bloodstream and move to distant organs. And a key cellular program that underlines this change is called epithelial-mesenchymal transition, or EMT. EMT is a reversible process in which rigid epithelial cells become mobile mesenchymal cell types. In metastasis, EMT allows tumor cells to invade adjacent tissues or get into bloodstream, which carries them to distant organs, where they can settle and proliferate upon mesenchymal epithelial transition, or MET. In laboratory, it's feasible to observe the effect of EMT with, for example, human mammary epithelial cell lines. And with one of these cell lines, we activate one of the EMT genes, ZEB1, and we tracked the nuclear eye of these epithelial cells. By comparing the motilities of these cells before and after EMT, we observed that activation of EMT greatly enhanced the movement efficiency of these cells. This shows the possible relationship between EMT and the invasiveness of cells. EMT is not invented by cancer. It's an essential step in early embryonic development, in which new organs need to be formed. This shows that EMT is an intrinsic process in many epithelial cells. In fact, it is controlled by defined gene regulatory networks of EMT. Shown here is a core EMT network. Blue icons are epithelial genes, yellow icons are mesenchymal genes, red arrows are inhibitions, green arrows are activations. With a mathematical model describing this complex network, we predicted that there exists multiple intermediate EMT states between the terminal M and E states. And these intermediate states can be stable. With human mammary epithelial cell line, we verified the predictions from the model. More recently, discoveries from other research groups show that in many different tumors, there exist these intermediate EMT states. And it's been proposed that in circulating tumor cells in bloodstream, there may exist these intermediate EMT states or cells. More recently, we started to ask how this core EMT network controls other EMT-related genes. In fact, it's been found that more than 700 human genes are related to EMT. Specifically, we focused on two core EMT factors, TGF-beta and ZEB1, and looked at how they control other EMT-related genes. We perturbed these two factors combinatorially by activating or inhibiting them and measured the activities of all other human genes, including these 735 EMT-related genes that are documented in literature. And with this high-throughput measurement, we used a machine learning technique called self-organizing map that assigns each of the EMT genes to one of these nodes. We first verified that this technique can reasonably separate the known E and AM genes. And we further looked at the classifications of mesenchymal genes. And we found that there are three major types of mesenchymal genes denoted as M1, M2, M3 categories here. And they have different connectivities to the core EMT networks. For example, the M3 group, which contains more than 70 genes, are primarily controlled by ZEB1, and the effect of TGF-beta is indirect. This is different from other M gene types, and most of the E genes are controlled by ZEB1 as well. Interestingly, we found that the high activities of M3 genes are associated with better prognosis of breast cancer patients. This is a surprising finding given the traditional understanding of EMT's role in cancer. This shows the complexity of the relationship between EMT and cancer. Currently, we're using new mathematical models and high-throughput measurement of gene activities combined with cell motility assays to gain better understanding on EMT, which ultimately will help us fight against cancer.